Thank you, Andy. I'm going to start this session. As you mentioned about combining Neuropixels acute recordings with optogenetics, I'm going to focus mainly about how to get started, what to plan when for your experiments, and how to prepare the animals for acute recordings and optogenetics. And Beau will explain more about what to do with the data and discuss artifacts related to the light. So this is just a quick introduction of the technique that we are talking about. Optogenetics, it's a method for controlling neuronal activity using the light. It was pioneered by Karl Dizerov and Ed Boyden, and allows us for very precise spatial and temporal control of neuronal activity. And right now we have many different options that can allow us to in controlled way, excite neuronal activity or inhibit neuronal activity. And optogenetics has a wide application to manipulate neuronal populations during behavior, and especially in combination with Neuropixels recordings. It also an amazing tool to map functional connectivity and also to identify genetically defined cells. So now I'm going to focus on some practical tips, how to get started your experiments and what you have to consider when you plan simultaneous manipulations and recordings. So the first decision that you have to make is how you're going to deliver the light, whether it's from inside or outside of the brain. And this decision will be determined by the option that you are going to use. So depending on your scientific question, you might choose either excitatory or inhibitory options, but all of these options are activated by specific wavelength of the light. And now the wavelength of the light also will determine how deep you can penetrate. So how deep, like let's say a blue light, 440 nanometers. It's good to use, for example, in the experiments that you manipulate cortical neurons. However, it's not going to be a good choice if you want to manipulate activity of neurons that are located much deeper in the tissue, because it's not going to penetrate so deeply. Whereas red lights like 650 nanometers will travel much deeper to the tissue. So if you want to manipulate activity of neurons, let's say in zona incerta or in thalamus, you might possibly consider using the red laser and manipulate the activity outside of the skull. And right now, because we have so many options and they are sensitive to, and they are activated by different wavelengths, depending on your experimental strategy, you can choose manipulating the activity either with blue or red lasers. And that applies to excitatory options and also inhibitory options. Another parameter to consider is the light source. So LEDs are cheap, but typically less powerful. So they can work in experiments in which you use very sensitive options that do not require so much laser power, and they will activate or inactivate cells reliably with little power. However, lasers are more expensive but more powerful, and they might be the only solution in case if you have options that requires a lot of power in order to activate or inactivate neuronal populations. And the almost last important parameter to think about, it's choosing optical fiber. So here what is important is a numerical aperture, diameter size, and tip shape. So all of these parameters will determine how the light will actually be scattered in the tissue and how big area you can manipulate. I will focus now only on a discussion of tip shape. Here you have example of flat fiber versus tapered fiber. And flat fiber, on average, allows to deliver like very local light in the tissue, whereas tapered fibers, you can see that you can reach much larger volume of the tissue. You can reach with the light much larger volume of the tissue. So that's something to consider and think like how big it's the brain structure that you are going to manipulate, whether you prefer to have very local and precise manipulations, whereas very big and bulk. And lastly, it's important to also think where you are going to position your probes, because that also will determine how you want to implant, for example, the optical fibers, if you will do a deep brain manipulations. And now I'm going to briefly introduce two preparations for acute recordings during optogenetics. First is when you deliver the light from outside of the brain, and this is typically for manipulations in the cortex, very superficially. It's less invasive. There's no tissue damage because the optical fiber is just positioned above the skull, above the craniotomy, and it gives you much more space because you don't need to apply cement around, and it's easy to record and stimulate from the same brain region. This is just an example of pictures of recordings in the visual cortex. Here you can see the Neuropixels probe going under some angle just below the optical fiber, and here how the light actually hits the region in this experiment. You can see some distortion, so you probably have impression that the light doesn't hit exactly the middle, but it's because there's some ACSF applied, so it creates some optical illusion. And the second preparation is when you decide to deliver the light inside of the brain, and that's typically for manipulation deep brain regions. In this example, this is superior colliculus, and then you need to implant the optical fiber deep in the brain, and that will result in larger tissue damage because of this preparation. It seems more invasive, but at the same time, it's more stable because you secure the optical fiber with a cement. So the optical fiber is always at the same position. And unfortunately, you have also less space for the probe, and recordings are more challenging, but it's not impossible to record. They are just more challenging, and you have to think about the plan. And this is example of the optical fiber implantation and simultaneous recordings from multiple brain regions during manipulations. And lastly, this is just to show you example of experiments that I perform using Neuropixels recordings and optogenetic inactivation of the left superior colliculus. Here, I implanted optical fiber just above the superior colliculus. The probe was labeled with a DIO and was going under some angle just below the area of where the expression of the opsin was. The opsin that I used was inhibitory opsin STGTA-CR2. It's labeled with red color. And these are examples of 4 clusters that have been recorded in the superior colliculus. And because this is inhibitory opsin, you can see that during light activation, the activity of these clusters increased comparing to control measurements without laser. But what was also striking in these experiments is that clusters that were positioned in cortex, they didn't express opsin, but they were also affected by the light stimulation. So in this case, there was some indirect activation of cortical neurons. And I wanted to just point it here that because Neuropixels probes are long, you can actually already record multiple brain regions only using one probe. And you can see what is the relation between manipulated brain region and other brain regions. And I'm not going to go through this slide. This is just a summary slide because we're going to share the slides with participants. And this is just a quick recap what to consider when you plan your experiments with optogenetics. So thank you for attention, and Maxime will take it from now.
Hello again. We're a bit late on time. Is it all right, Celia and Andy, if we carry on? Okay, cool. So let me share my screen again. Whoops, that's the other one. Yeah. All right, so again, I like showing raw data. here is some raw data from a Neuropixels probe, 300 channels over about, I think, 30 milliseconds, 15 milliseconds. And on this electrode was shined light between 0 and 5 milliseconds, which triggered an onset artifact as well as an offset artifact. And this can screw up your data, and you need to deal with this. We're going to talk now about technical considerations of how to pre-process your data acquired while you are doing optogenetics. So the physical basis of this artifact is the Becquerel effect or photovoltaic effect. You are ripping off electrons from metal when you're shining light on it. And the amplitude of this artifact is going to be proportional to the power, to the first derivative of the power of the light over time. So something you can do to mitigate it is using a smooth sigmoid as the onset of your artifact instead of a square pulse. But if your application is something like optotagging, you care a lot about the fast response of neurons to your optostimulation, and you don't want to use a smooth ramping photostimulation onset. So it's not always a solution. So in most cases, you have to deal with artifacts, and this is what it looks like if you focus on a single channel. And it's important to bear in mind that when you will pre-process your data, in particular, when you will high-pass filter it, you will make this artifact bleed out in the past and in the future because you are running your filter in both directions. You can read about ringing, which is a filtering artifact in this reference here that you can check later. But it's important to bear in mind because it means that it can affect your data in the future and in the past. The first issue encountered with artifacts is false positives. Bear in mind that KiloSort is an artifact picking machine, like it really is. If you run it on your data, it's going, for some reason, to co-cluster big artifacts together with your neurons. It probably has to do with the fact that the size of the artifact, if you do a dot product between a big thing and a spike, it will lead to a high matching score, and they will be co-clustered. We don't really know why it happens, but it does, and you need to be really careful with this. So here you see that it seems like this neuron was like fired after the optostimulation less than 1 millisecond later, about 0.3 millisecond later. That's way too quick, and that's not possible. The other issue that you have is false negatives, where spikes are masked by the artifact. And this is something which happens even more if you have saturation, which happens quite a lot on Neuropixels 1.0 probe. You can see here that the raw data is clipped right after the onset of the light artifact. That's a figure taken from our science paper. You can read more about it in the method section. And the solution is to pre-process your data and to do artifact subtraction. So you can do median subtraction across channels first, because if something is present across 3 millimeters of brain tissue, it's probably not a neuron. You can assume that. So you can just subtract the median across all your channels. You can high pass filter your data, and you can do spatial whitening, which is going to decorrelate the channels in space. So if something takes too much space, is over too many channels, is going to be removed, because again, neurons cannot be present across 1 millimeter of tissue. And this is all done in KiloSort. You can look at the details of this preprocessing pipeline in this very MATLAB script, but it's sometimes not sufficient, because if your artifact is homogeneous across channels, for instance, if you do wide field imaging, or if you do photostimulation in another brain region, so it's only scattered light which reaches your probe, you have a very homogeneous artifact across channels. And vice versa, if you are shining light from the craniotomy, you will probably have an artifact which looks very different across channel, which is what you saw earlier in the raw data. It will maybe be a little bit earlier and bigger on superficial channels than on light channels. In the first case, median subtraction does a good job, because again, the artifact is the same across channels, you can just subtract it off. But in the second case, it completely fails, and you need to do something else. You can harness another property of the artifacts, which is the fact that they will be consistent across instances, across light presentations. And so here in orange, you can compute the mean artifact, which will also be different across channels, and you can subtract that off the data, which will work already better. So here is an example of raw data. You have a heat map of voltage over time across channels, and you see if you only high pass filter it, you have a big ringing artifact, which you can see in red in the past and the future. if you just do median subtraction and whitening, which are the 3 steps of kilosource preprocessing, you still have quite a bit of artifact left. But if you do artifact subtraction, you're doing a better job. You still have some things left over here in the center, and that's why typically you would also remove the first, let's say 30% of the first millisecond of data, 300 nanoseconds, to be super safe. Something else you can do is demux scar, which is something which was invented by IBL, Olivier Winter at IBL, and implemented in SPAC-GLX by Bill Karsh. You see that channels are acquired in banks, and it leads to these kind of shifts in time of the artifacts between banks of channels. And so if you just do a mere median subtraction across these channels, it's not going to do a good job if your artifact is really quick. So what you can do is you can account for these differences in the time when the data is acquired within a bank of channel, which you know, because it's the same across all probes, and you can correct for it and do a better job at artifact subtraction. This is available on SPAC-GLX website. And then if you do a proper artifact subtraction, you will successfully remove the false positives from your rasters, but still look at your spikes, because false positives are the norm rather than the exception. Then the question that you ask yourself is whether your neuron is responsive. It fires way more spikes once the light is on, but is it some false positive response for some reason? So first of all, you want to know whether your firing rate is higher than some threshold when the light is presented, obviously, but it's really important to also consider the time when this increase in firing rate occurs. It should really be faster than 10, ideally 5 milliseconds, because I'm talking about experience. You have a lot of very delayed responses, whether they are behavioral, this will happen within 50 milliseconds maybe, or later, or whether they are caused by polysynaptic effect and reverberating activity caused by your optotagging. And glutamatergic synapses are not the only synapses which carry information across synapses. You also have metabotropic synapses, which will lead to delayed answers. So be really careful and make sure that before saying that a neuron responds to your optostim, make sure it responds within 5 to 10 milliseconds. And also use synaptic blockers, because we have examples of neurons which can be inhibited before gabazine, and when you put it on, the response completely disappears. That's quite trivial. Obviously, if you want to do optotagging, if you want to make sure that the neuron is directly responding to the light and not in a polysynaptic manner, you need synaptic blockers such as APV or NBQX or gabazine. Okay, I would like to acknowledge everyone. Thank you to Nick, Andrew, and this year Célian for organizing this course, Marius and Cyril, who are the Kilosort and Phi developers, Dimitar and Michael, who are my supervisors in the Horsa lab, and Tim for leading the Neuropixels collaboration overall. And yeah, thank you everyone.